Hi, my name is Alexander Shimabukuro-Vornhagen. I'm a member of the Laboratory for Tumor Immunology and Transplantation Immunology at the University Hospital of Cologne. In this video, we're going to show you how you can generate human CD40-activated B cells from peripheral blood mononuclear cells. One of the main interests of our laboratory is the study of the antibody-independent functions of B cells. Beyond their role as producers of antibodies, B cells have an important role as antigen-presenting cells, both under physiologic conditions as well as in autoimmune disease. After activation, B cells can acquire potent antigen-presenting and immunostimulatory capacities, especially when activated via CD40. CD40-activated B cells have several advantages when compared to dendritic cells. They can be easily expanded at relatively low cost from small amounts of peripheral blood, even from cancer patients. Now, our PhD student Tanja Liebig and our technician Fiedler will give you a step-by-step introduction on how to generate human CD40-activated B cells from peripheral blood.
Hello, my name is Tanja Liebig. I'm a PhD student and I want to demonstrate to you how to generate human CD40-activated B cells. This procedure was optimized in our lab. The main steps are: first, the preparation of feeder cells. Therefore, we use human CD40 ligand transfected NRH33 cells, which become irradiated with 78 gray and plated onto 6-well plates. Second, we start the culture of human CD40 activated B cells from purified PBMC.